Hello, this lecture is for Anatomy and Physiology 1 at Madison Area Technical College, and it will address an overview of the anatomy of the brain, the purpose and production of cerebrospinal fluid, as well as the structure and function of the blood-brain barrier. Further information can be found in your Merib textbook in Chapter 12, Sections 1 and 8. This also pertains to study question number 3. The average adult brain weighs about 3 pounds, and it's the size of both of your fists together. It forms from a hollow tube called the neural tube, which develops by embryonic week 4. So the picture on the left shows a blue tube with some outpocketings, and that blue area is hollow or space. As the brain develops, and it continues to develop in the fetus, the neural tube folds and forms outpocketings simultaneously. These outpocketings or swellings give rise to the 4 main regions of the brain that are present at birth and, of course, in adults. The 4 main regions are the cerebrum, the diencephalon, the brainstem, and the cerebellum. The brain is protected by the skull and covered by 3 membranes known as the meninges. The 3 meninges from superficial to deep are named dura mater, arachnoid mater, and pia mater. Please note in the picture that the dura mater has 2 layers. So the dura mater is 2-layered. Also notice that the arachnoid mater has spider-like extensions that secure it to the pia mater. The space between the arachnoid mater and the pia mater, where you see blood vessels, is called the subarachnoid space. This space contains cerebrospinal fluid that even further protects the brain. It cushions it, allowing it to float, and gives the brain buoyancy, which decreases its weight. This picture in the upper right shows 2 dural septa. So the dura mater's 2 layers penetrate in certain regions of the skull and form these partitions or septa. The 2 that are labeled are the falx cerebri, which is located between the 2 cerebral hemispheres, and the tentorium cerebelli, which is located in the transverse fissure between the cerebrum and the cerebellum. Another feature of the dura mater and its 2 layers relates to the fact that in some regions these 2 layers separate and allow the formation of large veins. These veins are called dural venous sinuses, and they collect venous blood from the brain to be returned to the heart through the internal jugular veins. In addition to being located in the subarachnoid space, cerebrospinal fluid is also located within the center of the brain. In the center there are 4 spaces called ventricles that develop from the hollow cavity of the embryonic neural tube. These hollow chambers are continuous with one another, allowing CSF to flow. 2 ventricles deep in each of the cerebral hemispheres are called lateral ventricles. They are separated from one another by a thin membrane called the septum pellucidum. Both lateral ventricles have a channel that connects them to the 3rd ventricle. The 3rd ventricle is very narrow, and it lies between the 2 halves of the thalamus. it is connected to the fourth ventricle by a canal called the cerebral aqueduct. So the fourth ventricle lies dorsal to the pons and the medulla oblongata. Its wall has 3 openings into the subarachnoid space called apertures, lateral apertures and median apertures. As I mentioned, the cerebrospinal fluid is located within the subarachnoid space and the ventricles. In addition, another location of the cerebrospinal fluid is in the central canal of the spinal cord, which is located right here. Functions of the cerebrospinal fluid, as I mentioned, include buoyancy, suspending the brain and reducing its weight in the skull. It also protects the brain from suffering impact injuries, but maybe most importantly, it regulates the chemical environment of the central nervous system. It's produced by the vascular choroid plexus, which is labeled on the diagram as the red tufts of capillary beds. And this is continuously produced. Cerebrospinal fluid is continuously produced, approximately 500 milliliters each day. But only 100 and 160 milliliters is maintained or kept within the ventricles and the subarachnoid space. That's the only amount that will fit. Therefore, a certain volume must be reabsorbed from the subarachnoid space into the dural venous sinuses, or the venous bloodstream. So the pathway of cerebrospinal fluid flow starts at the choroid plexus. Once the CSF has been produced in a ventricle, it flows inferiorly to the next ventricle. From the lateral ventricle, CSF would flow to the third ventricle, then to the cerebral aqueduct, and then the fourth ventricle. Any CSF that's produced in the third ventricle would continue to flow inferiorly and go directly to the cerebral aqueduct and then the fourth ventricle. When cerebrospinal fluid reaches the fourth ventricle, it leaves the interior of the brain through apertures that puncture the pia mater and allow the CSF to enter the subarachnoid space. As more and more CSF is produced, a force is generated causing the CSF to move superiorly, where there are openings into the dural venous sinuses called granulations. Once CSF has been reabsorbed into the venous bloodstream, it will return to the heart, proceed to the lungs to be oxygenated, and then pumped by the heart into arteries. This slide shows the pathway of cerebrospinal fluid produced in the choroid plexus or by the choroid plexus of the lateral ventricle. So from the choroid plexus or blood plasma, CSF is produced and enters the lateral ventricle. It then moves to the third ventricle, the cerebral aqueduct, the fourth ventricle, through the apertures in the wall of the fourth ventricle, and out to the subarachnoid space, where it moves superiorly until it encounters granulations or slits called arachnoid granulations, and then absorbed into the dural sinus, which is the dark blue. Eventually, the sinuses drain into the internal jugular vein and return the blood, including this reabsorbed CSF, to the heart. So the choroid plexus is very important for making cerebrospinal fluid. And to talk a little bit about how it functions, we first need to understand its structural components. It's made up of 2 specific parts. One includes the capillaries. They're called fenestrated capillaries because they have slits between the cells and in the membrane of the cells. These capillaries are quite permeable. So one component are these fenestrated capillaries. The second component are the ependymal cells. They form tight junctions between them and are relatively impermeable, allowing water to cross, but not a lot of other things can move without the aid of transporters. So the choroid plexus does a type of filtration, performs a type of filtration of the blood, where plasma leaks out of the capillaries and then must cross the ependymal cells. Water crosses freely, very little if any protein can cross, and ions are actively pumped across the ependymal cells. What does get filtered across has a similar composition to plasma, that is mainly water-based, but much less protein than plasma does, and a different concentration of ions. So glucose leaves the blood and enters the CSF, oxygen does, some vitamins, and some ions will enter the ventricle cavity. While at the same time the ependymal cells will pump wastes and any unnecessary or harmful solutes out of the ventricle and hopefully into the capillaries. Any kind of impaired cerebrospinal fluid flow can cause hydrocephaly, and sometimes this is due to the overproduction of cerebrospinal fluid, but more often it is caused by reabsorbing CSF inadequately. So hydrocephaly is defined as an enlargement of the ventricles, and the enlargement is due, it puts pressure on the brain, and the enlargement is due to the accumulation of too much cerebrospinal fluid in the ventricles. If for some reason the choroid plexus is overproducing CSF, that of course would increase the volume of the ventricles and put pressure on the brain, resulting in the condition hydrocephaly. The only case that I know of where CSF is overproduced is when there is some sort of a tumor in the choroid plexus. It's usually benign, it doesn't spread, but it would have to be removed. Other causes of hydrocephaly are more common, and they relate to the problems with reabsorbing cerebrospinal fluid into the venous blood system. That could include blockage or a tumor, which could cause blockage, I guess. Infection would cause swelling. Hemorrhage would mean that it would be difficult to reabsorb into a vessel, or some sort of congenital malformation that leads to blockage. The most common site of blockage occurs in the cerebral aqueduct because it's already so narrow. So the third topic today relates to barriers that help protect the brain and maintain the chemical environment that it needs. One that we've already discussed is the blood CSF barrier. That's only occurring in the choroid plexus, where the ependymal cells have tight junctions. When people use the phrase blood-brain barrier, they're usually referring to the tight junctions that exist in other capillaries that supply the brain tissue. So there are tight junctions between the cells of capillary walls in the capillaries that aren't part of the choroid plexus. So whereas the capillaries of the choroid plexus are quite permeable and the ependymal cells prevent the movement of substances, all other capillaries have tight junctions and form the blood-brain barrier. We'll look at some pictures. Hopefully that'll make more sense. First, let's talk about the arterial blood supply. that goes to the brain. An extensive network of vessels provide oxygenated, nutrient-rich blood to the brain, and the majority of the cerebrum is supplied by the internal carotid arteries. The vertebral arteries also provide blood to the brain. So the aorta branches into what is called the brachiocephalic trunk, which then gives off the common carotid arteries on both sides. The internal carotid artery branches from the common carotid, and as we'll see, it at the base of the brain, it splits into several smaller arteries. The vertebral artery travels from the subclavian artery, which isn't labeled, up to the base of the brain. If you look at the base of the brain, you can see that there are quite a few vessels that supply the medulla oblongata, the pons, the cerebrum, the midbrain, the diencephalon, and form what are known as anastomoses. That's a union of blood vessels that provide collateral circulation. So the paired internal carotid arteries and the paired vertebral arteries branch extensively within the skull, and at the base of the brain, some of their communicating branches form a circular connection called the circle of Willis. The advantage to having multiple blood vessels supply a region of the brain is to provide collateral circulation, ensuring that if one artery is blocked, the region of the brain will still receive blood. So the importance of the blood-brain barrier between these blood vessels we just looked at, what escapes them and what gets to the brain tissue, what the importance is to regulate and maintain a stable environment for the brain. Any chemical variations could cause a dysfunction or uncontrolled neuronal firings. So, like the barrier that we saw in the choroid plexus, the blood-brain barrier has specific components. There are three components. One is the tight junctions that exist between the cells of the capillaries. These cells are called endothelial cells. The second is that the endothelial cells are layered with a basal lamina, much like a basement membrane. It's a thick basal lamina, almost a connective tissue. And then finally, the third layer is the feet or podocytes of astrocytes that surround certain capillaries and neurons. So the advantages to this blood-brain barrier would be that it would prevent the spread of infectious microbes from getting into the nervous system. It would also prevent certain medicines, however, from reaching brain tissue. So that would be a disadvantage. Here's a picture of the blood-brain barrier. So this is a capillary that you might see on the surface of the cerebrum, not part of the choroid plexus. And the salmon-colored cells are endothelial cells, simple squamous epithelium, that are held together by tight junctions. Those tight junctions do not allow even water to pass between cells. Then surrounding the endothelial cells is what is called the basement membrane. It's difficult for hydrophobic and hydrophilic substances to cross that. And then surrounding the capillaries are foot processes of astrocytes, which are also connected to neurons. In order for a substance to leave the capillary and enter the brain tissue, it has to cross all three layers. So the blood-brain barrier and its three layers are impermeable to some antibiotics, certain drugs, so that can complicate the treatment of neurological diseases. However, it is permeable to water, glucose, iron, oxygen, and carbon dioxide. And that's due to partly, I know I said before that the tight junctions don't allow water to go through, but it's due partly because aquaporins are in place in the endothelial cells. It's also permeable to certain things that you're probably familiar with: alcohol, caffeine, nicotine, and anesthetics, because all of these do affect our brain function. And there are two drugs that successfully cross the blood-brain barrier, both for treatment of neurological disorders. Lithium is used to treat manic depression. L-DOPA is used to treat Parkinson's disease. So the blood-brain barrier is really regulated by the membrane proteins and the aquaporins that are present in the endothelial cells and the ependymal cells. Some treatments even use the ependymal transporters for substances to cross. That's all, and thank you very much for listening.